Francois and Betsy really set the stage for this part of this session. So we have unknown sequences. You're all familiar with BLAST, but there's more than BLAST in helping us understand where these unknowns fall. It's important for species delimitation, it's important for classification. So I just want to really briefly review what are the sources of unknown sequences. You're all familiar with metagenomics. This generates a huge number of sequence reads. And you may be aware that many of these have no close matches to anything in existing databases. And what happens is we just throw them out. It's convenient because we don't know where to place them, and there's no good mechanism or approach to place these right now. Also, there's sequences in public repositories, Treebased, Dryad. Many of these are poorly annotated. We heard Conrad talk about the need for looking at internal consistency, validation. We need to do that. And there's also a lot of unknowns generated through the Earth Microbiome Project. Betsy put up one of the PIs on that, Rob Knight. And you're generating a huge amount of data there of microbial diversity across the globe, but we're only using a small fraction of that. And John showed some of the work of NOAA with the alternaria distribution maps, but there's so much more data in there that we're not using. So we need to actually tap into that somehow. And of course, there's also cultures, and we heard of these large culture collections and herbaria. There's the opportunity to return to type specimens to understand more fully the species limits. There's, as John said, we could sequence genomes now. So this is extremely valuable, actually having specimens in hand. Okay, so how do we deal with these unknowns? How do we identify unknown sequences? The easiest thing is BLAST, but that's a pairwise thing. It doesn't care about anything about evolutionary history, but it's very fast, and it works well. You can detect perfect matches or sequence hits with low similarity. But what's really critical about BLAST and the utility of it is to identify these highly divergent sequences and to filter them. And the reason I point this out, this is important when we're doing alignment of reads to curated reference alignments in trees. Highly divergent sequences will throw off these algorithms, so we need a filtering step there. This is generally better than BLAST for aligning for alignable sequences. And the reason is it takes advantage of the shared evolutionary history within a clade, which we're ignoring when we're doing BLAST searches. It's problematic for unalignable regions. You have to be able to align the region. If there's site saturation, violations of position homology, which you would expect as you get more global in your alignment, then it's not useful. So there are limitations there. So how do you overcome those? You do reference-guided searches using BLAST. And this is something that Francois mentioned. And this improves the accuracy because now you could do your blasts to the unalignable sequences, which did not get into the reference tree and the reference alignments. But you have an anchor now. So think about ITS, LSU. The ITS is unalignable. It didn't get into the reference alignment, but you have the LSU. You have the 5.8s. So you could blast to the highly repetitive or the unalignable ITS and have your anchor to the 5.8, the alignable region in your alignment. And then you could focus on those specific clades. And this is what I'm going to talk about, the importance of not working globally, but once you've done these preliminary placements, to then focus on clades. So the approach is globally you do a blast search to initially place these sequences. Then you focus on the phylogeny, specific clades in the phylogeny for your identity. And this is more powerful because as you zoom in to a specific clade, then you have more information. For one thing, you're dealing with a shorter evolutionary time, so those unalignable sequences are now alignable. You have more resolution and more accuracy for placement of your unknowns. So, how do we do this? There's no tool out there that will actually allow us to zoom into a phylogeny and then say, I want to now examine all the sequences in this clade. So I want to do a local alignment, or I want to do a local analysis. They're all global. Most of you have used tree viewing software, Fig Tree, PhyloWidget, Archaeopteryx, but these tools, they don't allow you to actually store information, voucher data, or alignments right in along with the tree. So there is nothing. I would love to build on an existing tool, but there's nothing out there. And So we have to start from scratch. We have to reinvent or invent a new tool, and we did this. This is the Treebased Alignment Selector Toolkit. And I want to provide you with an overview of what it can do right now and where we want to go with this. So in the most essential feature of this is it allows you to really manage large multi-locus data sets that are not static but dynamic. They're really changing through time, and the user has to be able to continually update their tree, their alignments, their voucher tables. So you need the real-time ability, but also important, these have to be web-based evolutionary resources. And the reason for that is, imagine building a standalone tool. You'll never have the resources to map thousands of metagenomic reads, and if you have 1,000 users online, that's also going to be a drain on a server. So you want to take advantage of distributed high-performance computing resources that are online. Makes sense? And that's what we want to really plug into. So it's important that we do this online. And we want to be able to do the blast placement of unalignable sequences on the reference tree, then the phylogenetically aware placement, because we want to compare. Well, how did the two compare? And you want to see that visually. But then you'll also find cases where the alignment is ambiguous, no matter what you do. And so you need to have tools that will estimate the alignment or the uncertainty in the alignment and the phylogeny simultaneously. But you can't do this globally across all your alignments. It's just not computationally feasible. So by focusing in on a clade, then we could use tools like Balify to simultaneously estimate the alignment and the phylogeny and get very accurate placement of the unknowns in that clade. So this is a clade-specific approach to phylogenetic placement. Of course, we have other data. We want to integrate this. We don't want to lose track of the other, because really, to make sense of it, you need the genetics and the phenotype. You need to link these together. And you want to do it also visually, because there's nothing better than the human eye in seeing patterns, and then you could test those patterns for significance, again, on a clade-per-clade basis. So we want to integrate phylogenetic, organismal, environmental data. And we want to use standards that will allow us to not only share our results with other groups, but we also want to take data generated by other groups, Avatal, and we want to bring it into our toolkit. The holistic knowledge is important here. We don't want to be on a tangent developing something on our own. We want to be able to share these. So there are internet standards. There's XMLs, there's JSON. These are standards that are out there. Why can't everyone adopt those, at least allow export of their data into these formats to allow exchange between these different programs? So that's another objective, and that's really important because we don't want to be an island. We want to share our resources. Okay, so I want to walk you through and just show you some screenshots of what Treebased can do and why it's important that we built in all this functionality. So we need to allow for missing data among loci, and as Francois showed with that cumulative supermatrix, this is what typically the type of data you have. You have all this is you don't have data for these taxa, you have it for here, these, but I just want to add that this is typical not only of amplicon sequencing and what we're doing here. I'm a population geneticist. We use double digest RADseq. This is the type of data that you're getting. It's very patchy. You don't get restriction fragment data for some reads. And so this is something that moving forward, we need to actually be able to manage data with all these unknowns and allow us to keep adding. So when we have more data, we continuously add and update. We need to retrieve voucher data for specific taxa, and this is one of the functionalities that I think is very useful. Our approach is to integrate all the data into one large XML. This is a standard way of coding all the information so that you could easily retrieve it from a phylogeny. So this is the Treebased interface, and we wanted to have the most real estate for the tree in the center, and then have the options to the left and the right, and those dashboard windows there that give you information. As you click on those nodes, you see information within that particular clade. So you can go in here and select taxa, and those are highlighted. You can see them here. You can click on the node, and those are highlighted, and then you have this summary window here. It tells you the number of loci in there, the number of strains, taxa, and you have the option then to view or download your single alignments. So you could download single alignments for all the taxa in that clade. Oh, you want them in Phylop format, Nexus, whatever format you desire, you could download them. But more importantly also, you could download multi-locus alignments. You could do that in Phylop, but it's even more informative to do it in a format called Nexus, because when you download them in Nexus, you get this nice partition block at the bottom. And this partition block tells you where all the partitions are, all the different data sets that were brought together in the multi-locus alignment. Now, this is useful because when you're working within a clade, these partitions can actually specify unaligned regions that were excluded when you did the global alignment. So when you did the global alignment, you had to leave out these regions because they're unalignable. But when you're doing local alignments now you can bring those unalignable regions back into the fold. Suddenly you have a lot more resolution of relationships in that clade. So you see, by focusing in on specific clades, you have more power to place these unknowns. You get a lot of data and flexibility to pull or download information depending on how much you're willing to tolerate. How much coverage do you want for taxa and loci? So this shows you an example here. If you want data across all 5 loci, okay, you're only going to get 9 taxa with that. But if you say, at least 1 locus, you get all 82 that's in this clade here. So this gives the user control to download data sets where you can have information across all loci, a particular set. You may be interested in some taxa, some specific taxas. You want to know, give me as much information that I can get for these specific taxa. This will allow you to do that. You could download your voucher data for those taxa too, and this is in a CSV or an Excel formatted table. So alignments along with other information. So it's a one-stop place to get all the information that's available for those taxa. Even more important now, you could save this in XML format. This allows you to share it between programs. So this is built in. So not only the alignments in format that you could view them on your own viewers, but to share between tools and between different studies. Okay, so we want, of course, this is not a static thing. We want the user to have the ability to update. It's a moving target. You get more data, more taxa. You need to constantly update the tree, the alignments. And we allow the user to do this. On the right there are all what I've ticked off there are the reference alignments, reference taxa, all the files that are in a curated reference tree. So let's say you have your unknowns and you want to know their placement on our reference tree, you would select those options there. But let's say you have your own phylogeny, you have your own table of attributes, you have your own partitions, and you want to visualize them, you want to put them in this context to help you explore your data. You can do that. You can upload your Newick tree on the left, you can upload your partition, the data, your alignments, your tables, attribute tables, your unknown files, your FASTA files with all the unknowns or your metagenomic reads. So you could update. Okay, it's not static. Here's where it's really important. We can now look at placement of unknown sequences using multiple approaches. And one is blast placement that I talked about. And this is what I view as the top-down approach, or the global approach. So you could specify if you want to blast against a particular partition in the tree. And the reason I say this is because you may want to blast against the expanded ITS 5.8S LSU, which did not get into the alignment because you had to trim, but you could blast against that and then anchor those hits on the tree. So that's what you see on the second one, extended ITS 5.8S partition. We're actually blasting against that partition. So what that allows you to do then is anchor those hits on the tree, and on the outside ribbon here that you see, those sticking out, those are all the blast hits mapped onto the tree. So now we have them there. you could actually download them. Okay, so you could show the blast. Oh, these are just as an aside showing you that there's a lot of metadata that's output from this, and you have access to that. You will know what are the best hits for those unknowns, and this dashboard window provides all that. But there's also output that's provided that you could download. So this shows the best hits, and you could download then the sequences for that clade, for not only the reference alignment, but your blast hits now. So you have a file that, and I'm putting this, I'm specifying in FASTA format because it might not be aligned at this stage. You have to now realign it, okay? But you actually have all the best hits of those unknowns to a specific clade. Okay, you may want to use a phylogenetic aware placement using the reference alignment. So let me explain how that works. This is the workflow for evolutionary placement here. You start with BLAST, and that's to filter out those highly divergent sequences. And the reason is evolutionary placement has a problem with highly divergent sequences, so we need to remove those. You could use the BLAST anchoring approach that we use to place those. And then you could use a tool like Balify to further align them. But once you've blasted and you eliminated those highly divergent sequences, you use an alignment program called Papara. This was developed by the Stamatakis group, and it actually uses the curated the reference alignment and the reference tree to align your unknown sequences to the reference alignment. This is much better than not using the reference alignment because it uses that as a guide. Then once you have that, you got to trim the excess stuff on the edges that hangs out, and we can use G-blocks to do that. And then you could use this partitioned evolutionary placement algorithm that's provided in RaxML. And this is just showing you the output that we provide, the metadata of all these steps. So we give you the Papara alignment, the G-blocks trimmed, the blast hits that were not hit or were highly divergent for that partition and that are excluded. And so there's a list right there. So here you have the placement only based on the 5.8S partition, so the evolutionary placement. And you could see that big fan right here. That's the placement right there. And there's a number of others in here. Now, obviously, there's very little resolution here if you just base it on the 5.8S. But you could base it on the LSU. You could have multiple partitions that you're asking it to do your placements. So here, the majority fall within the Sordario-mycetes in the 5.8S, for the 5.8S only. Okay, so, but you could run it for the LSU and get even better resolution. So what if your sequences are really those blast hits, they're really divergent, and there's also the opportunity to use Balify or to simultaneously take into consideration that uncertainty in the alignment. And this is not yet implemented, but this is something that we want to do in future developments of this tool. And we're already set up to do this because you could select any one of these clades and download not only the alignments, but the blast hits that fall into that clade. Okay, and then finally, we have this integration with multiple data layers. So we want to visualize other information on the tree. And this is an illustration of how we do that. We do this with these outer bands. And you have on the left-hand side here the attribute table that you upload into, it's just a column Excel table. The headers become all these options that you can select, and they're automatically color-coded for you. And you could visualize different attributes on the tree. You could look at, let's say here, subphylum, class, and this really helps, although it's categorical right now, it's not quantitative. You could actually have quantitative data and do heat map distributions for that quantitative data on the phylogeny. So this is a way of bringing together all the disparate data parts that you have, all the information that you have for these taxa. This is key. We want this use internet standards, XML and JSON, to really to help with our data validation, but also to facilitate the data exchange with other systems. And here, for example, Avatal, you want to be able to share resources and share information. And the way we do it, we use PhyloXML. This is extensible markup language. I don't know if you're familiar with this, but this is an internet standard. This is something that's very commonly used. And so with this allows us to actually export our data and use it in other systems, and we have the potential to import other XML data into our tool. You might not be familiar with JSON. You may be familiar with JBrowse, these web-based browsers. They're all based on this file format called JSON. This is JavaScript. You may have used tools that are sometimes, have you used like Archaeopteryx or these tools that are applet-based. Very frustrating to use, these applet-based tools. There's no more of that now. Now it's all JavaScript, really enhanced that take advantage of HTML5. And this is a format that allows us to share our the visualization aspects of our tool. Okay, think of JBrowse, these visualization tool for genomics. You can have visualization tool for phylogenetics. And it's an interesting concept, but we could use that. You could practically share information between browsers. Something to think about. XML is an entry point to using these shared high-performance computing resources. There's no way that I'm going to put this up on my server, even with 56 nodes, and you'll come back and it'll crash if there's 100 people using it, right? You need to take advantage of high-performance compute. And these other organizations have done it. iPlant, Cypress, they've plugged into the Exceed high-performance computing resources provided by NSF, and these are available for everyone. You get an allocation. If you got more use, you just say, I need more servers, I need more. Okay. So we want to plug into our tools, not only Treebased, but our portal, into these high-performance computing resources. This will greatly accelerate, not only, it will help the public use these tools. It will be available, it'll be posted here on our website. So check under the resource tab, and when you see Treebased, you're ready to use it, all right? And with that, some acknowledgement on our funding sources, and thank you.